Many forms of cancer are genetic. Breast cancer is one of them, but until now, doctors were unable to predict which women in a family could potentially develop the disease. Now, an international team of researchers led by the University of Melbourne has discovered the first breast cancer gene, which can be used to predict which family members are at high risk. Professor Melissa Southey was in charge of the study, and she joins me now from Melbourne. So, Melissa Southey, is this discovery a real line of defence for women who have breast cancer in the family?
Yes, it adds to actually two previous genes that are currently used in clinical practice, but it certainly will allow us to identify women who are at high risk of developing breast cancer, and also perhaps more importantly identify women who are not at elevated risk of developing breast cancer.
And so women who do have this breast cancer gene that you've discovered, they will go on to develop the disease at some point?
Well, everybody actually has this gene. It has a function that's very important to our ongoing maintenance of normal biology. It's having a very rare mutation in this gene that causes the cancer predisposition. And no, not every woman who has this variation will go on to develop cancer, and this is consistent with the other genetic information that we have about breast cancer. So it's going to go on into very personalised risk assessment for women.
How did you find the gene?
It involved thousands of Australian families that participated in our research, and we applied new genetic sequencing technology to sequence massive amounts of DNA in these families to identify this gene as being mutated in families with many cases of breast cancer.
So you've worked out how to do it. When will these tests be available to women and doctors?
Well, the technology is currently being transferred into medical genetic services now. It'll be a little bit longer before we know precisely the elevation of risk to be able to tell these women very accurately what their risk is, but we hope it's going to be very short term rather than long term. We know how to handle this information now, and I think it'll be fairly rapid.
And could this technology be used to spot other cancers?
Yes, I think that's probably one of the most exciting things for us. This gene has been identified very early in a very large study, and we're hoping that other genes will be able to be identified very rapidly. It's also applicable to many other human disease situations, so we think this is really the beginning of a really big revolution in genetic health in a very broad sense.
Melissa Southey, thank you.
Thank you.